Hello all, this is Sahana presenting you Mendelian genetics. So all living organisms reproduce and results in the formation of offspring of the same kind. However, these offsprings may not be the same or may not totally resemble their parents. There are several characteristics that may differ between individuals belonging to the same species. So these differences are termed as variations. The mechanism of transmission of characteristics from the parental generation to the offspring is called heredity. Now, that being known, the scientific study of heredity, variations, and the environmental factors responsible for these variations is known as genetics. The word genetics was first suggested by William Bateson to describe inheritance. In genetics, there are different areas like classical genetics, which deals with the Mendel's principle, sex determination, sex linkage, cytogenetics, and so on. And there is molecular genetics that deals with the genetic material, its structure, replication, and expression. And there is evolutionary genetics that merely deals with the population genetics. Now, the history of genetics dates back to the classical era with contributions by Hippocrates, Aristotle, Pythagoras, and others, whereas modern genetics began only with the work of Gregor John Mendel in the year 1862. His work on pea plants established the theory of Mendelian inheritance or Mendel's Law. So, Gregor John Mendel is known as father of genetics since his work. So, Gregor John Mendel was known as father of genetics. He was born in the year 1822. He was an Austrian monk. He also worked as teacher, and he was fond of gardening and fruit culture since his childhood. So, while in teaching profession, he performed series of experiments on pea plant in his garden. So, his work contains inheritance of characters in 22 varieties of garden peas. And in the year 1866 and 67, he published the results of his hybridization experiments, which is titled as Experiments on Plant Hybrids, in a journal called The Proceedings of the Natural History Society of Brünn. And he postulated the principles of inheritance, which we popularly call as Mendel's Law. And he did a statistical study. He had a mathematical background, so he did a statistical record for each and every experiments that he conducted. He discovered that individual traits are inherited as discrete factors, which means discrete factors or genes, okay, which is later on called as genes. And these factors retain their physical identity in a hybrid or in a progeny. And the gene, it is a term which was coined by William Johnson in the year 1909. And he did his experiments for 7 long years, that is from 1856 to 1863, and he proposed his laws of inheritance. And he died in 1884 without any recognition. So why Mendel's work was not recognized? Though Mendel carried out his experiments for 7 long years on Pisum sativum, his work was largely ignored by scientists at that time, which was due to communication problem, and he used mathematics in biology, and the biologists got irritated, and new concept of genes or factors and something new as usual faces a great amount of resistance, and he couldn't present any solid proof for the existence of factors. And who gave him recognition? Later in the year 1900, the works of Mendel was independently experimented and rediscovered by 3 biologists named Karl Scorens, a German guy, Hugo de Vries from Holland, Erich Schemark from Austria. These 3 guys, they independently worked on Mendel's experiments and rediscovered Mendel's laws, and that's how Mendel got recognition. And why pea plant? Why Mendel choose pea plant? So Mendel chose the pea plants for his experiments since it had various advantages, like it is easy to cultivate, It has a relatively short life cycle, and it had well defined discrete characters. It had bisexual flowers, and it had a predominant self fertilization, and it was easy to hybridize. So characters studied by Mendel. The characteristics of an organism or a plant in this case are described as characters, or it's also called as traits. So the characters that was studied by Mendel were qualitative traits, that is the traits that are easily classified into different phenotypic classes like variations that can be visible. So Mendel chose 7 different characters or 7 qualitative traits, and they are as follows here. So the first character that Mendel chose was height, in which the dominant character was tall and the recessive character was short. And the second one is seed shape. The round seed shape was dominant, whereas the wrinkled one was recessive. And the third one is seed color. The yellow color seed is dominant here, and the green colored seed is recessive. And seed coat color, green color coat, seed coat is green, that was dominant, and the white seed coat color was recessive, white or kind of yellowish. And the pod shape is another character or another trait that was chosen by Mendel. The pod shape was inflated or full without any constrictions, that was the dominant character, and the pod shape with constrictions is called which is also called flat, is considered as recessive trait. And pod color, the color of the pod, green color was dominant and yellow color was recessive. And finally, flower position was another character chosen by Mendel. Axial position of flowers is the dominant trait, and the terminal position of flowers is a recessive trait. So before moving on to the topics, there are some terminologies that to be known. So the first one is factor. It is a unit or particle of inheritance or a gene responsible for the character. The things that was being stably passing down, which was unchanged from the parent to the offsprings through the gametes over successive generation, is called as factors. It was called as factors by Mendel. Later in the year 1909, Danish botanist William Johnson termed these factors as genes. So genes and factors are one and the same. Alright. So a gene can be defined as unit of heredity that influence the outcome of organisms' characters. Next, allele. Each allele may exist in alternative forms known as allele. Alternative form of gene is called allele. So whatever the alphabets that you can see here, those are the genes. These are the genes, and these two pairs are called alleles. These are alleles, P and P. These are alleles. These are alleles, alternative forms. And then here, each gene codes for particular character or particular version of character. For example, C and C, these codes for color of a pod or color of a seed. So these two code for same character but different version of that character. Say for example, this is for dominant color of a seed, that is yellow, and this is for a recessive character of a seed, that is white or green. And then homologous, it's a pair of chromosomes with the same gene sequence. These are the homologous chromosomes. This is one and this is one. This is a pair of homologous chromosomes. One is inherited from male parent and the other one is from female parent, and these has same gene sequence. As you can see here, the gene sequence are same on the both the homologous chromosomes. So the next one is dominant and recessive allele. So here I told these are alleles. This pair of genes are called alleles. So here this is a dominant allele. which mask or hides the expression of recessive allele. Say for example, this is a dominant color and this is a recessive color. When these two are present in a gamete, in separate gametes, and when they express themselves in the first generation, it's obvious that the dominant character is being expressed and the recessive character is being suppressed. So the character or the allele that expresses itself is called dominant allele, and the one which gets suppressed or is suppressed by the dominant allele is called recessive allele. Fine. And one more thing is, the dominant allele are represented by uppercase letter or capital letter, and the recessive alleles are represented by lowercase letter. And the next one is homozygous and heterozygous. Homozygous indicates they possess two identical alleles for a trait. So for example, this height is for axial placement of flowers. These two are capital A and capital A. That means both are dominant characters, and both these pair of genes are same. They are alike, so they are called as homozygous dominant. In this case, even here they both are same. Okay, so these two are recessive characters, but still they are same, so it is called homozygous recessive. Whereas in this particular pair of genes, one is dominant and one is recessive. In that case, they are not alike, though they holds the characteristics for particular trait. They are not the same. They are one is dominant, one is recessive, so they are called as heterozygous. Genotype and phenotype. So the genotype is defined as genetic constitution of an individual for any particular character. In this diagram, yep, here these are the genotypes: A, B, B, P, P. So these are the genotypes that holds that is a that actually passes the particular character to the progenies. And the phenotype is the observable properties of an individual for particular trait. It is a trait that is imparted by the genotypes. Pure line or true breed. Pure line is a parent which is in which the stability of character is maintained even after repeated self-pollination for several generations. They can also be called as true breeding parents, which means even after self-fertilization for several generations, they don't lose their uh, traits, and that is being passed on to the several generations. And progeny. Progeny is the next generation offspring, or the next generation offspring is called the progeny. And we'll come across first filial generation or F1 generation. So the progeny that is produced as a result of cross between two pure line parents or true breeding parents, that progeny or offspring is called as first filial generation. And F2 generation is the one, the offspring that is produced from the cross between two F1 hybrids. Whatever the F1 hybrids that is produced from two pure line parents, when that is being self-crossed, a progeny is produced, and that is called F2 generation. And we'll come across back cross. What is this back cross? It is a crossing of F1 progeny back with one of the parent. It can be a dominant parent or a recessive parent. Well, what happens in a test cross? In test cross, it is crossing of F1 progeny back with recessive parent. Okay, this is in particularly with recessive parent. And there is a definition called emasculation. Mendel used this emasculation technique in his experiments. So what is this? It is a technique used to bring about conditioned pollination, wherein the stamens are removed to prevent self-fertilization in cleistogamous flowers. What happens in cleistogamous flowers? It is a closed flower. So what happens in that case? The stamens should pollinate the stigma of the same flower. So self-fertilization is a rule there. But since Mendel wanted variations, he removed the stamens of the cleistogamous flower and he pollinated with the he pollinated that flower with the different stamen, the stamen from the different flower. So that is how he conditioned the pollination and he altered the self-fertilization technique there. So that's about part 1. we'll be dealing with monohybrid cross in the next part. Thank you.